Um, so my name is Alan Yesin, and my partner is Sonia Singh, and we are uh, presenting on characterization of electrodes for neural interface devices from the Integrated Electronics and Biointerfaces Lab, lab of Dr. Dye. And Sonia is going to get started in introducing our experiment this year.
So our project consisted of characterizing clinical electrodes of different diameters ranging in sizes of 10 to 800 micrometers, which were composed of the metals platinum, titanium, and polyethylene dioxithiophene, which is also known as PDOT. So to carry on this experiment, we had to understand the basics behind neural clinical electrodes, and these electrodes are probably smaller than the size of a dime. So these electrodes are placed directly on the surface of the brain, as seen in Figure 1, in patients suffering from brain tumors and other drug-resistant tumors. And what these electrodes do is record brain activity and pinpoint exactly in the brain where the brain disorder is coming from in patients. And for these electrodes to work, they require characterization regarding their electrochemical properties, which can be done by testing them in a saline solution to model how they would work in the brain. These characterizations consist of impedance, which is a form of resistance in the electrodes, which means that if an electrode has higher impedance, it would have more noise and is also considered unsafe in patients. And also cyclic voltammetry and charge injection capacity are other properties that were assessed, which are tests of when the reactions become reversible and also test how long the electrodes will last. So we tested these three properties, which are basically impedance, cyclic voltammetry, and charge injection capacity by setting them up in different configurations on the Gamry software, which is seen in figures 2 and 3. So that's what we did in our lab. And the goal of this project was to understand how diameter and the three materials we're talking about, how they affect the properties of the electrodes, which are essentially responsible for the safety of electrical stimulation to the brain. So moving on to the experimental design, we first characterized the clinical platinum electrode array seen in Figure 4. And from there, we were able to characterize electrodes of different materials and diameters, which led us to further test the electrode signal to noise ratio in a rat. And so the first step of characterizing the clinical platinum electrode array was to gain insight and practice how we can test the properties of different electrodes and also serve as like a control variable to make sure we were going on the right track of step 2, which was characterizing those electrodes of different materials. So for steps 2, we can see that in Figure 5, we tested this electrode ribbon, which was like made of 64 electrodes. And you can see in Figure 6 how these tiny multi-material electrodes really are. And Alan can talk more about the data that we found.
Yeah, thank you, Sonia. So looking at our experimental results from the initial step of characterizing the clinical platinum electrode array, we can see in Figure 7 our potentiostatic impedance measurements for platinum electrodes of various diameters. And what can be observed here is we saw that the impedance decreased as the surface area of the platinum electrode was increased. And then this is significant, as Sonia said, because lowering the effective resistance on an electrode allows us to obtain a lower signal to noise ratio, giving us more exact measurements or readings in a clinical setting. Then moving on to the animal case that we conducted, we essentially placed a novel electrode of various diameter clusters and various materials on the barrel cortex of a live rat's brain. And what we did was we fired equal stimulations of oxygen, or equal puffs of oxygen onto the rat's whiskers to essentially generate an electrical signal in the brain that we were then able to observe. And what can be seen from figure 10 is that we observed that the lowest resistance at microelectrode diameters, the impedance was the lowest for PDOT and the highest for titanium. And then moving on to figures 13 and 14, these figures essentially tell us that the average amplitude was lower compared to what our expected data was in figure 15. And this indicates that the data that we collected for the PDOT electrodes, there had been a significant reduction in noise as opposed to the other two materials. And then this is significant and basically leads us to our conclusion from this electrode characterization, in that the PDOT electrodes would, we would want to keep testing these in an animal case study settings and keep analyzing them, as these are the electrodes that we observe the least noise from. And like our conclusion would essentially be that these would be the most effective electrodes for some kind of clinical application because of the lower impedance and therefore lower signal to noise ratio. We would like to thank the Integrated Electronics and Biointerfaces Laboratory faculty and all of the graduate students that helped us with our experiment, especially our graduate mentor, Samantha Russman, and of course, Dr. Dye for all of the time that he put in to give us help and give us feedback on our poster. And we would also like to thank the GEAR program for organizing our lab placement this year. Thank you so much, everyone. And now we open to any questions.